Hello, good morning, everyone. Let's get to it. We'll be doing the test one review, and I just posted a link in the chat box for the review sheet if you haven't downloaded yet. And so the test is going to be on Thursday. Information, of course, is on eCentennial, and the test will cover everything from lecture 1 all the way to lecture 5. The review does not have material from lecture 1 though, but you know, it doesn't mean you don't have to study. You still need to go and review that lecture, but we will start reviewing from lecture 2 just because of interest of time. Okay, so first question here: what holds the atom together in a water molecule? Water molecule is considered as a covalent compound. Okay, water molecule is covalent compound. So what's holding it together is covalent bond, right? So just to refresh your memory, the difference between ionic bond and covalent bond is that the covalent bond involves sharing of electron. So if you have your water right here, H2O, it is going to be slightly negative on the oxygen and slightly positive in the hydrogen. And again, this bond here that's connecting the O and the H, we call that the covalent bond, okay, which involves the sharing of electrons. Okay, and for ionic bond, it would be a transfer of electron. And how would you describe the water molecule? Well, the covalent bond that we have over here has an uneven distribution. Oops, that's really hard to read. Uneven distribution of electron. The electrons hang out more towards the oxygen than the hydrogen. So the water molecule is what we call polar, right? Polar water molecule. H2O is polar. Okay, but important to distinguish the difference between polar and charged. Charged means you actually have extra electrons, or if you're missing electron. In this case, the water molecule has equal number of electron and proton. It's simply polar, meaning one part is slightly negative, the other part is slightly positive. But overall, the molecule is neutral. Okay, so no excess or missing electron. So of course, what happens is if you put two water molecules next to each other, right, one water molecule here, and then we're going to put another one right next to it, they will be attracted to each other. Okay, so the slightly positive portion of hydrogen will be attracted to the oxygen, right? And these interactions are what we call hydrogen bond. Okay, hydrogen bond. That's it. Moving on. If you supply radioactive nitrogen atoms, something that can be traced and detected, to a cell and let it grow, what molecules would you expect to find the radioactive labels in? This is basically like a fancy way of asking what molecules contains nitrogen. So of course we have 4 major macromolecules. We have the carbohydrates, which contains carbon, hydrogen, and oxygen. Right? We have proteins. Proteins also contains hydrogen, carbon, oxygen, but in addition, it also has nitrogen. And if you remember, it has trace of sulfur as well. One of the amino acids has sulfur in it. Lipids, we have CHO, right? And then we also have a phosphate on phospholipids. And lastly, we have the nucleic acid. Nucleic acid, so the DNA and the RNA, right, that has carbon, hydrogen, oxygen, nitrogen, phosphorus on them. So if we supply the cell with radioactive nitrogen, you would expect to find them in proteins as well as nucleic acid. Okay. Moving on. Regarding DNA, what is the monomer for DNA? Monomer for DNA, anybody? Want to remind me? What is the monomer for DNA? Anybody remember the monomer for DNA?
Nucleic acid.
Yeah, thank you. That is correct. Nucleic acid. Oh, sorry. No. That's actually not correct. DNA is a type of nucleic acid, but the monomer, the building block for DNA, is actually the nucleotide. Nucleotide. Good try though. Okay, so nucleotide. What is a nucleotide? A nucleotide has three parts, right? Nucleotide is made up of the sugar, right, the phosphate, as well as the nitrogenous base. Okay, so these three things forms a nucleotide. Okay, so the sugar could be in the case of DNA, this is a deoxyribose sugar. Deoxyribose. For RNA, it will be a ribose sugar. So this one is missing an oxygen compared to the RNA one. Phosphate, as you know, is PO4, 3 minus. And nitrogenous base. What are the possible nitrogenous base for DNA? Anyone? What are the possibilities for the nitrogenous base? Yep, there is the adenine. Very good. Thank you, Mavis. Anything else? Guanine, yep. T and C, cytosine and thymine. That's right. There's no uracil though, Sandeep, because we're talking about DNA. But you'll be correct if we also include RNA. Okay, so uracil is found in RNA and not DNA. So there we go. Those are the nitrogenous base. So how are nitrogenous base classified? You might remember we can either classify them as pyrimidines. Pyrimidines. So pyrimidines are T and C. They have single ring in their structure. Okay, so it looks something like this. I mean, you don't have to draw them. I'm just drawing it to show you. Right. So there is a single ring structure right here. That's a pyrimidine. And on the other hand, the other classification is that it could be a purine. Okay, purine would have 2 rings, and it will be A and G, double ring. Okay, so something looks like this. Okay, we'll have 2 rings in the structure. And so a pyrimidine will pair with a purine all the time to maintain consistent length along the DNA, consistent width rather. Okay, so what is the base pairing rule for DNA? T pairs with A, and then G pairs with C. Okay. What force holds the nitrogen bases together along the length of the DNA? And so if you think about a strand, this is the backbone, right? The DNA backbone. DNA backbone, which is made up of sugar and the phosphate. Right? Remember, the nucleotide has the sugar, has the phosphate, and then it has the nitrogen base. So the backbone of the DNA is made up of entirely just the sugar and the phosphate. And you're gonna have your base sticking out like this. So if I if I know one side of the strand, I will be able to predict what's on the other side using base pairing rule. Right? So what holds them together is the hydrogen bonds. Okay, so A pairs with T, there are 2 hydrogen bonds that holds them together. Whereas for C and G, there will be 3 hydrogen bonds. Okay, excuse me. Okay, so these are hydrogen bonds. Okay, 2 between A and T, 3 between C and G. Right, so it's a stronger hold between C and G. So what's the significance of the base pairing rule? Well, if you remember, the A and G, these are purines. Okay, purines. And the C and the T, those are pyrimidines, right? So pyrimidines over here has single ring, whereas the purine, it has double ring. So if I take a purine and pyrimidine and I bond them together, this is always going to be 3 rings wide. Okay, we want to maintain a consistent width along the entire length of the DNA. Okay, so this allows us to maintain consistent width along the entire length of the DNA. Okay, you don't want some parts to be 2 rings wide and then 3 rings wide and then 4 rings wide. If you don't have a consistent length, A is difficult to package, and number 2, it might create instability in the structure. Does anybody have any questions so far? Okay, no questions. Let's move on then. What is dehydration synthesis? Well, you probably is familiar with this concept now. If we take individual monomers, okay, monomers, this is monomers, we can connect them to form polymers. Okay, so the process involved here is dehydration, synthesis. Okay, you have to remove a water molecule, right? So plus H2O if you want to join them together. And if you want to break it apart, then it's going to be hydrolysis. Yes, William, I will post this after class. Okay, so there we go. Again, what connects the monomers together? Probably should draw it better here, right? So these bonds that hold them together, those are covalent bonds. All right. So example, let's put some actual example here. So monomers, we can have amino acid, and then after you connect them together, you would have a protein, right? Protein. If we have, let's do another example. If we have nucleotide, then we would end up having like a DNA or RNA. Okay, So that's the relationship between the two. All right, and we did a question in lecture 2, right? If I have, let's say, a 6 carbon ring structure here with an OH, and then we add it to a 5 carbon ring structure, OH, and I say, let's connect them together through dehydration synthesis. What's going to be the product? Well, the product is going to be removing the OH here first, and then removing an H to form the H2O. And in the end, you are going to get this guy, right? Of course, plus H2O. So some of you might remember we did a question like that. If you have not seen this question on any of your quiz yet, probably we'll see it on the test. Okay, so there is the ring here for the 6 carbon ring, and the other part is going to be the 5 carbon ring. And basically this part here is going to connect with that, right, to form the bond. Okay, any questions? Okay, moving on, regarding enzymes. Okay, so we have a bunch of enzyme questions here. What is an enzyme? Anybody want to tell me what an enzyme is? Help me out here. What is an enzyme?
Enzyme are protein.
Yep, they are proteins. Very good. Enzymes are protein. Yep, and they are not hormones though, Hema, but good try. Hormones are proteins though. So both enzymes and hormones are proteins, but enzymes are not necessarily well, they're not hormones. So what does enzyme do, huh? Anyone else want to give it a try? What does enzyme do? Yeah, it hastens reaction. That's pretty much it. Okay, so enzymes are proteins. Okay, they catalyze. Catalyze means speeds up, right? Chemical reactions. I hope it's not too late to mention this, but even though the test is all multiple choice, when you study biology, you really need to write stuff out, okay, to help you remember them. If you just read the notes or read the PowerPoint or just watch the recordings and whatnot, that's not going to have the same effect as writing things out. Okay, writing things out actually does help you remember it a little bit better. Okay, so they catalyze chemical reactions in the body by lowering, and that's what William is saying, lowering the activation energy. Activation energy. Okay, so what is activation energy? Let me just review that for you. So in a chemical reaction, okay, on the y-axis we have energy, and on the x-axis we have time. So your reactants, that means your starting material, it's going to have an energy level here, okay? And in order for them to react, right, we must supply a little bit of energy before it's going to react. So this would be the products. Okay, so the amount of energy that is supplied here, that's called the activation energy, okay, EA. Energy required to kickstart a reaction. If you remember what we learned last time with the metabolism, right? Like glycolysis, for example, which is a bunch of reactions, but consider as a whole, you need to supply some ATP first, right? So that could be thought of as activation energy before the reaction goes on and produce more ATPs. Okay, so this might be a little bit too much to supply in a biological system, and so with the help of enzyme, we are able to start at the same point, but now we supply less activation energy. So EA required with help of enzyme is lower. Okay. Next question. What is the next question? The next question is, what factors affect the functionality of an enzyme? Anybody? Yep, very good, Gorinda. This is a protein, right? As many of you mentioned. So protein is very sensitive to various factors that would affect its shape. If you change the shape of a protein, you affect its function. So we basically are thinking about factors that would affect the shape of the protein. So we have things like temperature, right? If you heat it up too much, it will denature, right? pH, that's good. Concentration of what though? That is a good one. Concentration of what? Yeah, of salt, right? Of ions, for example, right? Okay. Types of solvent, right? What is it dissolved in? Example, is it a hydrophobic, hydrophilic, hydrophobic, right? If you put a protein in a hydrophobic environment, it actually will flip inside out, and of course it won't work anymore. Okay, very good. Something like this could be like a multi-select question. You can imagine how that goes. What does the enzyme lactase do? Okay, lactase is an enzyme that breaks down lactose, and the way you know is you look at the suffix, right? ASE is an enzyme, and then that tells you what it breaks down. So lactase breaks down lactose. So it takes lactose, and in the presence of lactase, what will it become? What is lactose made up of anyways?
Glucose and galactose.
That's absolutely correct. Thank you. So it will break down into glucose plus galactose. Okay, these are, of course, monosaccharides, and that would be a disaccharide. Right? So obviously you should know the other ones as well, right? There's maltose, right? And then there is fructose. So make sure you, sucrose rather, sorry. Make sure you know what makes up what. Okay, great. Moving on over here. Let's make this a little bit bigger. Okay, carbohydrates, right? So what are the monomers of carbohydrates? Well, I kind of just talked about it just now, right? We have the monomers, are monosaccharide, right? The single sugar, right? So examples of monosaccharides include glucose, right? We have fructose and galactose. Okay. Biological functions, what do we use them for? They are primary source of energy, right? So an example of that primary source of energy would be glucose, right? You know that by now, right? This is burned, metabolized in glycolysis, cellular respiration, right? What's another biological function of carbohydrates? Anybody? What else do we use carbohydrates for? Helps in the formation of a structural component. Yep. Structural support. right? And components, and that would include things like cellulose for plant, cell wall and fibers, or chitin, which would form the exoskeleton of various organisms with shells. Okay, another one is storage of energy. Okay, storage of energy, which would be starch for plants, and for humans it is glycogen, which we store in muscle, for example, as well as the liver. Okay, there we go. Moving on. Proteins. Okay, so proteins, the monomers for proteins are amino acid. Okay, amino acids. There are 20 different amino acids, right? 20 amino acids. 8 of them are essential. You need to eat them. Your body cannot make them. The other are non-essential. So function for proteins, structural support again. Structural support. For example, like your nails, horns. We don't have horns, but like, you know, some animals do. Hair, some proteins in it. Muscle. Okay. We can use them as fuel for secondary sources. So amino acids, depending which one it is. Okay, but amino acids can be converted into various intermediates in cellular respiration. So you know there are many steps in cellular respiration. So like the glycolysis has 10 steps. So some amino acid can be converted into something that would feed into like step 6 or something. And then the Krebs cycle has many steps as well. You can convert some of them into those intermediates as well. So you know, then you can metabolize that from that point onwards and produce some ATP. What's another function of protein? Proteins. Help me out here, please. It's my last class with you, so participate a little bit. Another function, please.
Enzyme.
Yeah, we talked about that just a little bit earlier, right? So they can form enzyme, thank you, which would, again, catalyze reaction, right? So an example of that would be lactase, okay? Energy source, yeah, fuel, right? Energy source, okay? And then another one is a transport, right? Remember, there are so many membrane proteins function, right? So one of them is transport, and that would allow us to like move things across the membrane. And then another function would be like hormones. Okay, hormones. An example of a protein hormone is insulin. Okay, remember, there are lipid hormones as well. Okay, but insulin would be an example of a protein hormone. Okay, so that's that. Lipids. Let's see what we've got for lipids here. Remember, lipids are a little bit different compared to the other ones because they don't have monomers. Instead, they have like components. So things like glycerol, right? We can have some fatty acids, which could be saturated or unsaturated. And you know, you can have a phosphate group as well. Okay. So depending which one you're talking about, the function could be, for example, a major component major component of cell membrane. And that is, of course, phospholipid. Phospholipid bilayer, right? Cholesterol is another key component of the cell membrane. All right. Another function is that they will insulate and protect organs. And the lipid that does that would be the triglyceride, which is just a fancy word for fat. It could form water-resistant surfaces. That's would be the job of wax. And as I mentioned earlier, we could have steroid hormones as well. Okay, so the sex hormones, the testosterone, the estrogen, those are steroid hormones. And of course, we can also have storage of energy. Okay, and that would also be triglyceride. Okay, give you a moment to catch up. All right, nucleic acid. The monomer of nucleic acid is nucleotide, as we discussed earlier. And the function is it contains the the instruction to make proteins. Right, that would be the function of DNA. Right? Like, how do we know what amino acids to link together? Well, it's all encoded within the DNA. Right? It is, of course, also the hereditary material. Which is, you know, what you passed on to the next generation. Just give me a second here. I'm gonna reply a quick email. Sorry. And another one is it is used by ribosome to make proteins. Used by ribosomes to make proteins. Okay, and that would be RNA. All right. So there, that's your macromolecule chart. You know, in a very brief summary. Okay. Excuse me. Allergies is still going crazy for me. Okay, next question. How is the shape and the structure of a protein stabilized? So this question talks about the 4 levels, right, of organization for protein. So we start with the primary structure. Okay, so the primary structure is we take the individual amino acids and we connect them together, right? Okay, so this is called the primary sequence, which essentially is the sequence of amino acid. Okay, so if you read each of these amino acids, that order, that's the primary structure. And what determines the order? The DNA, right? The DNA has the instruction. Exactly how it does that, you will learn it in the second half of the course. Connecting the amino acids together, okay, so that's the amino acid. Connecting the amino acid together would be the peptide bond. Okay, so that's the primary structure. Next level up, we have the secondary structure. So if you take this long strand of peptide, they will curl up together, and some parts might form these helices, and some parts might form these zigzag patterns. They might have only helix, only zigzag, doesn't have to be both. But whether they form the helix or the zigzag, we call that the secondary structure. So you can have alpha helix here. We can have the beta pleated sheets. Okay. And the key thing to know here is that the secondary structure is stabilized by hydrogen bonds. Hydrogen bonds formed in the peptide backbone. Okay, so I just got a question, a private message question. Okay, I'll get to it in a second. Okay, but let's finish this first. Okay, so that is the secondary structure. And after secondary structure is, of course, our tertiary structure. Okay, so for tertiary structure, it is going to be this thing folded up into a 3D shape, right? Okay, so like you might have the helix here, helix, and then it just folds back on itself. Right, this is like a 2D thing for the most part. It's like flat on a piece of paper, but now you crunch it up and make it into a ball, then you get your tertiary structure. So this is referring to the overall 3-dimensional shape, okay, which is called conformation. That's a fancy word. And it is stabilized by interactions of the side chains, right? Remember, each amino acid has a side chain. If you don't know what I'm talking about, I'll show you in a second. And there are different interactions, okay? So let me give you some examples. Ionic bond, right? Hydrophobic interaction. That means like things that are hydrophobic would tend to clump together, so on and so forth. There's a whole slide on this. And that's tertiary structure. And of course, the overall 3D shape is what gives the protein its property, right? Okay, last but not least, we have the quaternary structure. Not all proteins are going to have the quaternary structures. Okay, only some will do this. This is when multiple polypeptides come together to form a complex. And an example of something that has this would be the hemoglobin, right, that we talked about. Okay, so there you have it, the 4 levels of protein structures. Any questions? Okay, I received this question in a message. Let me just post it here. Which of the following is a reasonable explanation for why unsaturated fatty acid helps keep any membrane more fluid at lower temperature? Oh, okay. Well, I don't see any options here, but I can tell you why unsaturated fats helps make membrane more fluid. Okay, so unsaturated fatty acids, they have kinks in the structure, right? Because of the double bond. So if you have several unsaturated fat next to each other, then they are not going to pack close together easily. Okay, and instead, what happens is there's more gaps between the molecules. Okay, so there's more gaps, basically, more gap, because the kink in the tail prevents them from packing together too closely. So if you have a membrane that has more unsaturated fat, it is going to be more fluid. Now, normally speaking, if you're talking about say a plant that grows in normal temperature, we don't want to have too much unsaturated fat because it's going to make it very leaky. Okay, do you understand that? If you have too much unsaturated fatty acids under normal condition, the membrane would be too leaky because it's too fluid. There is too many gaps in between, and things just leaks out of it very easily. However, if we have a plant that grows at low temperature, for example, then it's okay. In fact, it is probably a good idea to have more unsaturated fat, because otherwise, the low temperature would cause the membrane to be too solid, too rigid. Right? Think about putting butter into the fridge, right? It's going to become solid state, right? But if you increase the unsaturated fatty acid content in that butter, it's going to be able to remain relatively fluid, right? And so, as an adaptation for those cells that are or plants that are growing in low temperature, you would expect their cell membrane to have more unsaturated fat. Okay, does that answer your question, or is there like a follow-up question to that? All right, good. So conversely, for something that is growing in like a very hot environment, like the desert or something, you would expect them to have more saturated fat in the membrane and less unsaturated fats, right? So think about that for a moment. Because in a hot environment, the membrane would be too fluid, and you know, having additional unsaturated fat would actually make it even more liquid, and we don't want that. So we would have less unsaturated fat and probably more saturated fat. Okay, over here, identify the molecules. What is this one? This first one right here. That is, if you recall, that is the amino acid, right? Amino acid. Okay, and the amino acid has an amino group, and there is the carboxyl group. Okay, so when you connect the carboxyl group of one amino acid with the amino group of the other, you get a peptide bond. Right? And what about this? What is this? Anyone? Anyone recognize what this is?
Nucleic.
That is the nucleic acid. Very good. But it's specifically, it is a nucleotide. Okay, so very good. That's the phosphate right here. We have the sugar here. And there is your nitrogenous base. Okay. Over here, that is a glucose. And this one, fatty acid. Is this a saturated fatty acid or unsaturated fatty acid? Which one is this? That is saturated. Very good. And we can tell because there's no double bond. It is bond to a maximum number of hydrogens, right? No double bond within the chain. This one doesn't count. Okay, this one is for the carbon and the oxygen. Yeah, so this is saturated. Excellent. Moving on. Oh, that's it for lecture 2. Last call. Any question for lecture 2 before we go on to 3? We still have 3, 4, and 5. All right. List the major components of cell membrane. I mean, we certainly have done this before, but let's go through it again. We have the phospholipids bilayer, right? Okay, so we have the phospholipid bilayer, okay, and the hydrophobic tails are in between, whereas the hydrophilic heads are on the outside, right? Now, this is not a static structure. I have to emphasize that. Okay, so let's say if you focus on this red one right here, this is able to move left and right. Okay, so like if you take a snapshot of the membrane here, then you try to take another picture like a couple minutes later, the red one might move all the way over here. So like they are able to shift back and forth. Okay, it's not like a static structure. And in between the phospholipid bilayer, every now and then you're gonna maybe have a protein that goes through it, right? So that would be an what we call an integral protein. Okay, integral protein. An example of an integral protein would be a channel protein. Okay, so in transport, we learned that some molecules are not going to be able to go through the membranes. So let's say I have a charged particle, it cannot go through the phospholipid bilayer, right? Just like the H plus in the crystate, it cannot go through. So it's going to have to go through a channel protein. It forms a little tunnel in between for the charged particle to go through. So that's an example of facilitated diffusion. I'm just going to draw a few more of these things. And then another example of integral protein is you might have a carrier protein, right? Okay, so it will grab onto something and then it will change the shape, right? It grabs onto something, change the shape so that it opens on the inside and move the object across. Okay, now not all membrane proteins are going to be integral proteins. Some membrane proteins are just going to be stuck to one side, and we call that peripheral protein, right? And they might do things such as catalyze a chemical reaction, right? They might play a role in cell-cell recognition, forming a junction, for example. And they could also attach to things like cytoskeleton. One last thing that's missing here in our membrane is the cholesterol. Okay, so there's your cholesterol, and also these cholesterol are not static. They are able to move laterally, and cholesterol helps maintain the fluidity. Okay, and what crosses the membrane easily, right? Small non-polar molecules can cross the membrane easily. Okay, whereas if it's charged or if it's hydrophilic or polar, then it would probably require some kind of transport protein. All right. Moving on. Describe the central dogma of biology, the central theme of biology. So we have our cell right here. This is the cytoplasm. We're going to have our nucleus right here. Of course, our nucleus is going to be surrounded by the membrane, right? Somehow that does not look white. Probably because it isn't. Anyway, I was trying to make it look like this. Right, there are these nuclear pores. And so what happens is in the nucleus, we have our DNA. We have our DNA. Okay. And DNA is going to stay within the nucleus. We cannot allow it to leave the nucleus because it's too big, first of all, to fit through these pores. And secondly, even if we're able to get it out, we don't want to because it might get damaged. So when we need the instruction in the DNA to make a protein, we're just going to make a copy of it in the form of RNA. Okay, so this process is called transcription. Okay, so from DNA to RNA is transcription. And then from RNA to protein, when you use the instruction in the RNA to make the protein, it is translation. Okay, so this strand of RNA is going to exit. It's going to exit through the nuclear pores. And when it gets to the cytoplasm, you're going to have the ribosomes that clamps onto it. And it will convert, it will read the instruction and use it to make proteins. Okay, so these little green things, those are the polypeptide. If you're confused about what polypeptide and proteins, like how are they different? Polypeptide is basically just the primary structure, right? Before it folds up together, this still needs to create a 3D shape to form the protein. So that's the central dogma, central theme of biology. Okay. One of the functions of the liver is to remove toxin from the blood. Based on its function, which two organelles would you expect to be abundant in the liver? So we would expect it to have a lot of smooth ER, for example. Okay, and that's involved in detoxification and breakdown of lipids. Okay, what's another structure that would be abundant besides smooth ER? Let's do 3 here. What's another one besides smooth endoplasmic reticulum? Yep, there is the peroxisome. Very good. Peroxisomes, okay, which uses oxidative reactions to break down toxins and other molecules. And don't be scared by these big words. Oxidative reaction. We talked about oxidation last week, right? When something is oxidized, it loses electron. So basically, the peroxisome break things down by ripping electrons from them. Okay, much like glucose is broken down into carbon dioxide, right? When you rip electrons away from it. And one of the side effect of this is it actually generates peroxides, H2O2, hydrogen peroxides, as a byproduct. Okay, one more thing that you would expect to find a lot of in the liver cells. What else is responsible for breaking down toxins? Lysosome, thank you. That's right. Okay, so lysosomes. So this one contains mixture of enzymes, right, for breaking down. Again, toxins, macromolecule, and even ingested particles, right? Like a bacterium or stuff like that. All right, that's it. Structure of the mitochondria and state of function of each part. Yeah, we can certainly review that. There is the outer membrane. Right, and the inner membrane is folded up into the cristae, right? And the space here is the intermembrane space. Okay, and the function of the intermembrane space, as you should know by now, is that it allows the buildup of H+. Okay. Last week we learned that embedded in the cristae are these chains, right? These electron transport chain. There are many of them. I just draw a few here. Okay, and the function of the electron transport chain is to harness the electron's energy from the carriers, and it will pump H+ from the matrix into the intermembrane space. Okay, so the intermembrane space allows you to have this buildup of H+. Okay. And in order for them to come back into the matrix, they will have to go through the ATPase, which is what this thing is, this little doorknob thing. So the H+ goes through it, and in the process, you're going to be able to generate ATP. Okay, so this, of course, is the matrix. All right. So what else happens in the matrix? Let's write it down here. For the matrix, there are two things that happens, right? There is the pyruvate oxidation, step number 2, and Krebs cycle. And for the cristae, that's where the ETC is embedded in. Okay. So there you have it, the different parts of the mitochondria and its relation to the cellular respiration. Okay. All right, next part. Describe the endomembrane system. Okay. So we did this before, but let's go through it again. Here is your nucleus. And the nucleus is surrounded by the nuclear envelope. Okay, so there is the nuclear envelope, which of course you would know by now. Oh, where's my Y marker? It's going to have these pores on them, right? Okay, so all these gaps that I'm drawing here, right? Those gaps, those are the nuclear pores, which allows things to go in and out of the nucleus. So the nuclear envelope is actually going to be continuous, right? It's going to extend into the membrane, into the cytoplasm, and we are going to have this whole network of membrane. And some of them will have ribosomes on them, right? And some will not. Okay, so this here, this portion that has the ribosome on them, that would be the rough endoplasmic reticulum. Okay, and the function is that it works with ribosome to synthesize proteins, right? To synthesize and we'll say package proteins. And over here, this stuff that does not have the ribosomes, we will call them the smooth endoplasmic reticulum. Okay, so in addition to breaking down lipids, they are also responsible for synthesis of lipids, synthesis and breakdown of lipids. Okay. And further out, we would have another structure here. It's like a stack of membranes, right? And this guy is going to be the Golgi body, right? The Golgi body or Golgi apparatus. And the function is to sort, modify, and repackage vesicles, proteins. Okay, so you might have a vesicle that's coming along from here, right? There's a vesicle, and then it will fuse with the Golgi. And you know, go back and forth a couple of times maybe, and finally you will repackage it into a new one. Okay. So where will this go? Well, we talk about a couple of destinations, right? For example, it could be released immediately. Out of the cell, right? Okay, we're gonna get rid of it right away. It might become a membrane protein, embed into the membrane. It will be stored for later use. It might go to the lysosome, right, and become part of its proteins, right? Okay, so those are all the different destinations. Okay, that is it for lecture 3. Any questions about lecture 3? This is not an exhaustive list of questions, right? Like it's a good starting point, but you still have to maybe go back to the other study guides to have a more comprehensive revision yourself. Okay, so don't think this is the only stuff that you have to know. Okay, let's take a quick break. Okay, we'll resume in about 5 minutes. Okay, so see you at 9:40-ish. And we'll do review for lecture 4 and 5. Let's keep going. Lecture 4, explain what passive transport is. Provide example of passive transport. Okay, so passive transport is basically the spontaneous movement of substance, spontaneous movements of molecules, substances, doesn't really matter, whatever you're moving, from an area of high concentration to an area of low concentration. Okay, very importantly, no ATPs are required. Just happens on its own. So tons of examples that we talked about in class. Please give me some examples. Anyone? Yep, there's a simple diffusion. Very good. Um, protein pump requires ATP. So like sodium potassium pump is an example of protein pump. So that would not be passive transport. Facilitated diffusion. That's right. So that would be the channel protein or carrier protein. Osmosis is another one. Very good. So these are examples of passive transport. Talking about osmosis, don't forget to review this question. There is the U-shaped tube, right? There's a semi-permeable membrane at the bottom, and then you have all these big circles that represent the solute. Okay, so they're too big to cross the membrane, and then you have water level like this, same level at the beginning, and then over time, what's going to happen? So a couple key things to know here. Because the membrane is semi-permeable, the solutes will not be able to move. So the only thing that's going to be able to move would be the water. Okay, so water is going to move from left to right in this case, and then it will cause the water level to drop on the left side, and the water level, and the water level on the right side will rise. And at the same time that this is happening, the concentration on the right side will be diluted, whereas the concentration on the left side will increase as water moves to the right. Okay, let me just explain this one more time in case you weren't paying attention, because you probably will see a question like this on the test. Oh boy, there we go. Okay, this is really weird. It's not letting me move it. Let me try one more time. Okay. So over time, what's going to happen is water level will drop here and water level will rise here, right? Okay, so this side drops, becomes more concentrated, okay, because you have less water, right? But the solute did not change. And over here, the water level rises and less concentrated, gets diluted because the water is moving from left to right. Okay, so water moves to the right. All right. Hopefully you understand that. And if not, feel free to ask a question. Okay, bulk transport, different types of bulk transport. Bulk transport means to move large quantity across the cell membrane, and we can move them in or out. So this is exocytosis. Right, so remember that freshly packaged vesicle from the Golgi, right? Where is it going to go? Well, you're going to have the membrane here, and then you're going to have the vesicle right here with its content. This will move towards the membrane. It will touch it and fuse with it. Okay, like that. And then afterwards, it will completely become part of the membrane and all of its content will be released. Okay, so all this stuff will come out. And so that's exocytosis. The opposite of that is endocytosis, which basically is this thing, but reverse. You're gonna have some stuff you wanna bring in, it's gonna cave in on the membrane and it becomes a vesicle like that. And there are 3 different types of endocytosis that we talked about. There is the phagocytosis, which brings in solid. There is the pinocytosis, which brings in liquid. And then lastly, we have the receptor mediated endocytosis, where it is more selective about what you're bringing in. So, there's your membrane again, but this time you're going to actually have some special receptors on the surface. Okay, so whatever you're trying to bring in has to fit the receptors. So like these circle things, if it comes touch the receptor, nothing will happen because they don't match. But if we have these triangle things, right, once it binds to the receptor, it will trigger the surrounding area to cave in like that, along with the receptors, right, and the triangle stuff. And afterwards, what's going to happen is you will completely bring those things in. Okay. These are sequence of events that are happening, right? In case you're wondering what I'm drawing. And then the membrane will be sealed off, and then you would have a vesicle here with the little triangle stuff that we try to bring in. Okay. So the difference between receptor-mediated endocytosis and regular phagocytosis, for example, is the fact that you need to match the receptor first. So there is some selectivity in terms of what you're trying to bring in. Okay, you get to choose what to bring in, whereas this is more of a non-selective process. You might end up bringing things in that you don't want. Okay, so those are the different types of bulk transport. Of course, bulk transport is a type of active transport, requires ATP. Okay, next question. A cell of a plant grows in very cold environment. What kind of adaptation would you expect the cell membrane to have? We actually talked about this earlier when someone was asking that question, but let's go through it again. So generally, you would expect the adaptations will increase membrane fluidity. Okay, because in a cold environment, the membrane would tend to become more solid, right? So you need to counteract it. You need to make the membrane more fluid. And there are several ways to do this. We mentioned already. You can increase the cholesterol content. Okay, you can increase the percent of unsaturated fat. And you can decrease the percent of saturated fat. Okay, all of which will increase the fluidity of the membrane, which is what you want when it's too cold. You want to prevent it from becoming too solid. If it's very solid, nothing gets passed through, right? Okay, active transport, how are they similar to facilitated diffusion? So similarity. Both require require a membrane protein. Okay, so if you think back to transport like protein pump, that's going to need to have a membrane protein. And for facilitated diffusion, you need a channel protein or carrier protein. Okay, so that's similarity. And for differences, active transport requires ATP, facilitated diffusion, no ATP. This is from high to low, sorry, active is from low to high concentration. And then this is from high to low. Okay. That's it for lecture 4. Okay, next one. What is an organic molecule? I feel like this might have been something that we talked about even in lecture 2, but doesn't matter. Organic molecule is any molecule that contains carbon. Okay, except for carbon dioxide, carbon monoxide, and cyanide. All right, so whoever came up with this rule, they were like, no, let's exclude these things. I think there's a question on the test that asks you which of the following is an organic molecule. So choose everything that has a carbon in it, except for these ones. List all the products of glycolysis. Okay, so we start with glucose. We're gonna have to put in 2 ATP. We're gonna have to supply some ADP and some phosphate, and we're gonna have to supply 2 NAD plus. That's what we're gonna put in. Okay, so inputs. And after glycolysis, we're gonna get 2 pyruvate. We're gonna get 2 net ATP, and we will have 2 NADH. Okay. So these are the key products. Okay. In cellular respiration, glucose is oxidized while oxygen is reduced. So basically what that means is glucose will lose electron while oxygen will gain electron. Okay, so there's a transfer of electron from glucose to oxygen. So electrons are removed from glucose, picked up picked up by energy carrier, NAD plus and FAD, and ultimately transfer to oxygen, which is the final electron acceptor, right? So talking about energy carrier here, what is the role of NADH and FADH2? These are energy carrier, electron carriers, which is the same thing in our case because these electrons are high in energy, so they're electron carriers that captures electrons released from glycolysis, pyruvate oxidation, and Krebs cycle. The electrons are deposited onto the ETC. Okay, and just in case you've forgotten, NAD plus the electron will become NADH. Okay, I never told you where the H comes from, but it doesn't really matter. Okay, just the key point to know here is, you're picking up the electron. So this way it is reduced. Okay, because it's picking up electron, and at the ETC, it's going to go the other way around, which is going to be oxidized. Oxidized at ETC. Okay, and this would be reduced at, various steps, 1, 2, and 3. Okay, same thing for FAD. So you must know when is it reduced, when is it oxidized. Okay, next one. What is the significance of adding coenzyme A to pyruvate during oxidation? Just to refresh your memory, there are 3 things that happens to the pyruvate, right? So glucose becomes pyruvate, 2-pyruvate. This is glycolysis. And then the 2-pyruvate, we're gonna remove carbon dioxide. Okay, for 2-pyruvate will happen twice. We're gonna have 2 NAD plus coming along and becoming 2 NADH. That's gonna go to the ETC. But by this point, the molecule is kind of low in energy because we have been creating so many ATPs from it. We have been, extracting electrons from it. So it needs a little bit boost. It needs to be re-energized. And that's why we're going to add the coenzyme A. Okay. And so that will recharge the particle, right? Re-energize. We'll just say energize. Energize the molecule. So that is going to be ready to react again. Okay, so that will create 2-acetyl-CoA, okay, which will then head off to the Krebs cycle. Okay, Karina, they gain electrons as in the NAD plus, right? Not the pyruvate. Okay, just want to clarify that. Okay. Electrons are being removed from the pyruvate, and those electrons will be captured by the NAD plus. All right, math question. We did some of these last time in the tutorial, so let's try one again. Assume that mitochondrion contains 50 NADH and 19 FADH2. How many ATPs can we produce under ideal condition? Okay, what do you guys think? Give me some numbers here, please. Well, let me know when you have it. We have 188 from Mavis. Okay. Pashal said 150 from NADH, yeah, but together, how many? All right, very good. So NADH is 50, and we times 3, which gives me 150. right? Because each NADH can pump enough hydrogen to generate 3 ATP, whereas the FADH2 can times 2, which gives me 38. And if we add them together, that's how we get the 188. Very good. Next question. The inner mitochondrial membrane area is 5 times the area of the outer membrane. What is the purpose of this? Well, the inner mitochondrial membrane is where you have the ETC embedded in them, right? So by having a higher area, you are able to embed more ETCs, right? So this allows more electron transport chain to be embedded within the cristae. This will increase the amount of H plus pumped, which means you will increase ATP production. Okay. Which step of cellular respiration generates the greatest number of ATP? That will be the electron transport chain. Most of the ATPs are generated here through oxidative phosphorylation. Okay, as opposed to substrate level phosphorylation, right, which only happens in glycolysis, that gives you 2, and Krebs cycle, which gives you 2. Right? So 4 out of the 36 is generated through substrate level phosphorylation, and then the remaining 32 is generated through oxidative phosphorylation. Describe what happens to H plus. Sorry, describe what happens as H plus is pumped from matrix into the inner membrane space. We talked about this already, right? You generate a gradient of H plus in the intermembrane space. They cannot come back in, so they have to go through the ATPase, which creates the ATP. And finally, the purpose of the final electron acceptor is we also mentioned this multiple times last week, so I'm just going to tell you verbally. The final electron acceptor, the oxygen, is going to pull the electron away from the last protein in the ETC, so it doesn't clog up the whole chain, right? Because as we know, if you clog up the chain, we won't be able to deposit additional electrons, and we won't be able to generate the gradient, and therefore we won't be able to make ATPs. Okay, so there you have it. That is a review for the test. Does anybody have any additional question regarding the test before I talk about the lab?
Hello, Professor.
Yes, go ahead, please.
Our next midterm, it will be multiple choice. Am I right?
Yeah, so here you can see this is the test, right? 25 multiple choice, 10 multi-select questions, right? Multiple correct answers, and then 15 application questions.
Okay, thank you.
Yep. This is 20% of your grades, right? Everybody, and it's from lecture 1 to lecture 5. And it happens on Thursday. You can just click on the link and do it that way. Okay. All right. So like I said, this is the last week I'm going to be with you. And if you click on the weekly schedule, you will see it has been updated. I spoke with Professor Moran, and he pretty much is going to be doing the same thing as same format as me. You can see over here, you will have the asynchronous on the Tuesday and then synchronous on the Thursday, pretty much like what we did. Now, this week is the test, right? I told you. So where are we right now? You will do the test on Thursday. There will be no class. And next week you will watch the new recording post by Miran, and you will attend his synchronous tutorial. And in addition, you will also have to hand in the beetroot lab next Thursday. Okay, so I'm gonna talk about the lab right now. So please pay attention. So how do you get to the lab? Well, I will post a link today as well. Like after class, I will do my debrief and then I'll give you a link to the lab. But here is how you get to it. Okay, content, virtual labs, and then this is the beetroot experiment. Okay, so you can read this little blurb here, and there is a video demo. Okay, so if you open that, it will take you to a YouTube video, and you basically just have to watch that. Okay, so this is like a person who did the beetroot lab. I wanted to film my own, but then I never had a chance to go back to the campus to film it, and I found it online. So it's it's someone else doing it, but it's the same thing that you would have done if you were doing this in class. Okay, so you watch the recordings. Here is 8 minutes long. It's very important. I cannot stress the importance of this. You cannot do the lab just by, as they say, you cannot just wing the lab. Okay, you cannot just do the lab without watching the video. A lot of the questions, the answers to those questions is actually discussed within the video. And so what many people did in the past is they went online and they searched for the answers, and then they just put the answers into the lab rather than watching the video and getting the answers from the video. What you search from online is not necessarily going to be applicable, because what the person is doing here is different than the labs that you will find online. So rather than just, scouring through the internet to find the answers, just watch the 8-minute video and then you will have all the answers. Okay, it's very straightforward. So once you've watched that, you will download the lab. Okay, you might want to download the lab first. Okay, so you should click on that, and then here is the lab. Okay, you want to click the download button. I'm gonna do that, and then it will show up in your computer. Okay, so I'm gonna open that, and hopefully it's coming. There it is. Okay. So what you want to do is read everything, please. Okay, so review the lecture on cell membrane structure and transport. That's lecture 4. Watch the demo video, the one that I just showed you, and then you have to complete all these questions and submit it on eCentennial. Have you guys had a chance to submit stuff on eCentennial yet? Did your other teacher show you how to do that in other courses? Please let me know. Yes?
Yes.
Okay. Yeah,
Yes.
thank you. So it's the same thing. Okay, there is a folder. Sorry, I just gotta blow my nose. Hold on a second. Sorry. So it's the same thing. I'll show you where the folder is in a bit. I'll have a link to it as well. Okay. And what you're gonna do is you literally just type the answer in here. Okay. First question: what is the purpose of this experiment? Okay. Try to limit the answers to 1 or 2 sentences. Don't write us a novel. Okay. Just keep it short. All right. This is something that needs to be concise and correct and clear. So what is the purpose of this experiment? I'll give you the answer for this one. Okay, you will say the purpose of this experiment is to investigate the effects of temperature on membrane permeability. That's it. That's your 2 marks right there. Okay. I give you permission to copy this, okay? Don't try to be fancy and then use thesaurus and change the words or something like that. Just put this exact same thing, and you will get your 2 marks. I will communicate. I'm not the one who's marking it, okay? But I'll tell Miran to give you 2 marks when you have this answer. Okay, and then you go on to do the next one. What is the hypothesis of this experiment? Remember, the hypothesis is a statement, okay, not a question. In the past, people lose a lot of marks when they write a question here instead. So the hypothesis should be you will think the temperature will either increase the permeability or it will decrease the permeability, or it will have no effect on the permeability. Okay, state the independent dependent variable. That should be very straightforward. This one is another one that people get a little bit confused about. Why is beetroot chosen for this experiment? Okay, so why not choose something else? Why not choose carrot? Why not choose tomato? Okay, I mean, beetroot is red, and tomato is red, right? Carrot is orange. How come we don't use those things for the experiment, right? So you have to think about that. Safety precautions. Try not to repeat yourself. Some people say wear goggles, and the next one is like wear gloves. Next one is wear aprons. All this is the same thing. Okay, it's basically like wear PPE, right? Personal protective equipment. Think about the actual type of precaution that you have to take when you're doing this lab, in person. This question asks you what is a control of an experiment. Can someone remind me what a control is and why it is important to have a control in the experiment? We'll talk about it as a class right now, so you're not gonna get it wrong. Okay, help me out, anybody. What is a control? Why do we need a control? Do we need a control for all experiment? Same lighting. Okay, but in general, what is a control though, right? Not specifically to this experiment per se, because you guys haven't watched the demo yet, but I'm saying, like, in general, why do we
It's
need
the same some circumstances like atmosphere, the war flask used for experiment.
Okay, so what you are talking about is actually a standardized variable. Okay, so same circumstances, same setting, right? That's something that we have to make sure for all of the test tubes here, for example. Okay, so that's a standardized variable. But what about control? What do we need a control for?
The one we control, the one we know what the gradient or what we can control it.
Okay, yeah, so we're getting somewhere. Okay, so the control of the experiment is something that we have control over. Yeah, I mean, we actually can control the other ones too. We can control their temperatures, right? Okay, but I think you're getting onto something. And Mayfis here is giving an answer is used for comparison. Exactly, right? The control is the one that we are going to compare it to. Like, if we don't have the control, and then we see like red colors in all these tubes, how do we know what it normally looks like, right? So here, right, write this down, right? The control of an experiment is necessary for us to compare all the other treated groups with. Okay, so it gives us a baseline, a reference to compare all the other treatments to, right? So if we see a difference, we know is because of the temperature change, for example. Okay, so that's what a control is. Now, Karina mentioned standardized variable. Again, standardized variable is important, but it is not the answer for this question. Okay, this question is not asking about standardized variable. This question is asking about what is a control. Okay, so a control is the one that is not being manipulated, is the one that is not being treated, right? So room temperature. And the significance of having that is to have a reference so we can compare it to it. Okay, so what needs to be taken into consideration when cutting the beetroots? Again, the answer is in the video, but I am going to remind you, when you do this question, think about standardized variable. Okay. Sandy, maybe you can watch the recordings afterwards for that exact phrasing. Okay, but I hope I have conveyed the core idea of what a control is. And you don't have to write it exactly the way I've said it, but make sure you understand why we need that. And other than that, I think this is another one that people have trouble with. In the video, when you watch it, you will put it in the water bath for 20 minutes. Make sure explain to Miranda why do we do it for 20 minutes? Why not we do it for 10 minutes? Why not do it for 30 minutes, right? What's the consequences of that? If I do it for 30 minutes, is it going to affect the results? Or if I do it too short, is that going to affect the results, right? You must explain that. And qualitative and quantitative, people tend to have confusion surrounding that. So in this experiment, how was the result measured quantitatively? These answers could be found in lecture 1. Okay, there is a slide for quantitative and qualitative if you need to refresh your memory on it. Describe the relationship between the absorbance measured by the spectrophotometer. So I realized that in the video, the person actually does not use this word. They don't say spectrophotometer. They use the word colorimeter instead, which is the same thing for our purpose. But I just want to mention that. Okay, so COLOURIMETR, colorimeter. Okay, it's something that measures the color intensity. Okay. Some people spell it without a U, right? It doesn't really matter. So that's what this is asking. And then here, this is to me, this is like give away 4 marks very easily. Okay, all you need to do is plot this data. Yeah, this data plots the absorbance versus temperature. Now, in order to get 4 marks, you need to have proper labels on the y-axis, on the x-axis. You need to have a title for your chart, and you need to choose the right type of chart. Okay. Again, the person in the video actually did a graph, so just make sure you watch the whole thing, and you will know what graph to use. I spoke with your computer teachers, and as she told me, you guys already learned how to graph on Excel. Is that correct? Can someone confirm that for me? Yes. Okay. So that's where you can use the skill to do this. If you need help, right? Maybe contact your computer teacher, or there's a link here that would show you step by step how to graph on Excel. If you don't have Excel, you can use Google Docs, which is free. And actually, as Centennial students, I think you are entitled to a free copy of the Microsoft Office. So you can log on to your My Centennial account and access the let me just show you where to get that, because I think some of you might be interested in doing that. So if you go to your My Centennial, not your eCentennial, your My Centennial, there is the Microsoft eStore. Okay, you should have a link. Oh, well, it's very unfortunate that it is not available right now, but when it does become available again, or you can contact the help desk here, you actually can download a copy of it because you're a student. Okay, but yeah, you can use Google Docs in the meantime. Let me see. Beetroot test. Yeah, and the rest is pretty self-explanatory. Okay, I think it's an easy lab, and people who generally watch the video and do it tends to get good marks. It's the ones who leave it till last minute and then they try to find answers from the internet. That's when they don't do so well. Okay, I think I might have some examples of like. Give me a moment. Let me see if I have some of these things. Okay, so I have some examples here, and I'm not trying to make fun of people who make mistakes. I'm just trying to show you what you should not be doing. Okay, so over here, this is a graph that someone gave me. It's incorrect, right? You'll get 0 for this because there shouldn't be 2 lines. You should have temperature on y-axis, temperature on x-axis, and absorbance on y-axis. So this is not good. Let me see what else is not good. Yes, here, you shouldn't be handwriting the lab. Okay, I'm pretty sure typing would be much easier than writing the answers like that. Okay, so just type directly into the Word document and email the answer to me. And over here, This is another one. Someone copied something from the internet and forget to change the font. Okay, so I know that they have copied it from the internet. Just use your own words, guys. No need to plagiarize. Here is another graph that is very wrong. It shouldn't look like this. Okay, you should look at the one from the video, like I said, and find out what is expected. And here is another terrible graph. These are actual examples from student submission, right? Okay, like there is virtually no graph here. Okay, so yeah, that would give you 0 as well. So that's your lab. All right. Does anybody have any question?
Sir, this lab assignment, we have to do it after the test or before test?
You can do it anytime. It's available right now, but the due date is next Thursday. Okay. Hima said, do we need to make graph? Yeah, I was telling you, right? There was one question here, right? Below some spectral photometry data, you have to take these data, make the graph, label the graph, and paste the graph here, right? Okay.
Next Thursday means June 23rd.
Next test.
No, I'm talking about the lab test, beetroot.
Lab test. There's no lab test, right? Is there a lab? What lab test? Let me see.
No, I'm talking about the beetroot lab.
Yes, it is due next Thursday, which is the 23rd. That's right.
23rd, yeah.
Yep.
Okay.
Open now. We can do it now if we want.
Yeah, you can do it. You can do it now. You can submit it, but we it's not going to be graded, right?
Yes.
Yeah.
Yeah.
In fact, that's a great idea, right? If you finish your test, studying your test, and you just want to get it out of the way, honestly, this thing, if you sit down and do it, it's only going to take you an hour, right? And it's 10% of your grade, right? So might as well get it out of the way. Anything else?
We have a chemistry exam too in the same day. I'm worried that because we have to study both in the same day, we have a lot of information.
Same day as what? As as the bio test?
Yes,
Yes, we have 2 tests that day.
Yeah. So I unfortunately, as per college policy, 2 tests is okay. Is not something that we would change for. But I know it's a lot of stuff, right? Like I understand that. But unfortunately, I cannot change the day. Yeah.
Okay.
Yeah. Sorry about that. Okay. Well, I will still be available if you need to contact me for the rest of the week. But after that, like I said, Maren will take over. And I want to thank all of you for your commitment and participation in the tutorial. I will be back in September, and I will be teaching the second semester anatomy course. So maybe we'll meet again. But if not, I wish you all the best, and I hope you have a fantastic rest of the semester.
We hope to see you again in September too.
Yes, you could choose to enroll in my class again if you wish to. So, okay, thank you. Take care, guys. Bye for now.